it became necessary for crime labs to analyze every single cannabis sample that came in. And the reason is that the government decided that if you had 0.3% THC by mass in your plant material, if you had that level and lower than that, you had hemp. And if you have higher than that, you have marijuana, which means that to properly classify materials that are coming into the lab, you really have to quantify the amount of THC. Now, crime labs are very good at that. They've been quantifying THC for a long time.
Hello and welcome back to the Pittcon podcast. In this episode, we'll be speaking to Rabi Ann Musah about the challenges of differentiating hemp from marijuana in the lab. So thank you for joining us here today.
It's great to be here.
Great. So could you start by introducing yourself and sharing a bit about your background and what led you to focus on the analysis of cannabis evidence in forensic science?
My name is Rabi Musah, and by training, I'm a natural products chemist. And I'm particularly interested in the secondary metabolites or small molecules that are made by plants, how they make them, what they use them for, and how understanding of those secondary metabolites can enhance the quality of life for humans and animals, and the various things that we can use these small molecules for. So it turns out that some years ago, we were very interested in psychoactive plants in a forensics context. And we specifically were interested in how crime scene analysts can identify psychoactive plants when they're trying to solve crimes. So it turns out that there are over 400 plants that'll get you high. And in any particular country, a very small subset, usually less than 20, are scheduled or are outlawed. And so because of that, people import these plants with impunity in order to try to circumvent the laws associated with drugs that are in fact scheduled, like marijuana, for example. And because there are no laws that have outlawed these challenges, these drugs, what ends up happening is people overdose on them, and or they become they experience psychosis, negative health impacts. And it's very difficult for the judicial system to deal with it because there are no laws associated with those. Part of the reason for that is that you really need some sort of way to identify definitively these plant substances. And when people grind up roots or leaves or something like that, it becomes very, very difficult to identify them because they've lost all their morphological features. So we began by developing this approach where we take advantage of the fact that the genomic differences between plants are, of course, species specific, and that translates into species specific changes in metabolome or small molecule profiles. And so if you apply statistical analysis processing to the data that you get from the profiles, you can actually readily differentiate species. The challenge with that approach is that it's been difficult to do it quickly, and it's been difficult to build these databases of chemical signatures for plants. And so we found an approach using a very fast mass spectrometric-based analytical technique to accomplish that, and that really has helped us a lot. So that's direct analysis in real time, high resolution mass spec, which I'll refer to as DART-HRMS. And it turns out that very, very rapidly you can get these chemical signatures. So we realized shortly thereafter that could be applied to resolving some of the questions associated with cannabis. And so since then, we've been applying some of what we've learned with identification of psychoactive plants more broadly, specifically to cannabis.
Brilliant. That was so comprehensive. Thank you. So in your Pittcon talk abstract, you discussed the challenges of differentiating hemp and marijuana in forensic laboratories. What are the key factors that actually differentiate hemp and marijuana?
So it turns out that this is a challenging problem because hemp and marijuana are actually simply varieties of the same species of plant, Cannabis sativa. So while people have done genetic testing and it's clear that there are genetic differences between the two, as I said, they're the same species. So it's not feasible, however, for crime labs to use genetic testing to differentiate between the two. But one of the ways that they can be distinguished is in terms of the amount of the psychoactive component that's present, which is delta-9 tetrahydrocannabinol, popularly known as THC. And it turns out that while both hemp and marijuana have this component, in marijuana, it's a lot higher. And in hemp, of course, it's a lot lower. And so this is the main distinction. There are other distinctions, but it's the distinction that the government cares about because the Schedule I status of marijuana is directly a consequence of the higher amounts of THC that are present.
And what are the current challenges and limitations which make this differentiation between these two varieties in forensic labs so difficult?
Well, it turns out that because in the United States, at least, cannabis was outlawed, has been outlawed for a really, really long time, and it didn't really matter whether it was the low THC form, which is hemp, or the high THC form, which is marijuana. However, hemp around the world has traditionally been known as an agricultural product. It's very, very useful, actually, and there are lots of products that can be made from it. And the American farmers wanted access to that lucrative market. And so in 2018, Congress, the U.S. Congress, passed what's known commonly as the Farm Bill. And what it did was it made it so that hemp was an agricultural product. It used to be once upon a time that as long as you had cannabis, it didn't matter whether it was the hemp variety or the marijuana variety, it was outlawed. So when that happened, it became necessary for crime labs to analyze every single cannabis sample that came in. And the reason is that the government decided that if you had 0.3% THC by mass in your plant material, if you had that level and lower than that, you had hemp. And if you have higher than that, you have marijuana, which means that to properly classify materials that are coming into the lab, you really have to quantify the amount of THC. Now, crime labs are very good at that. They've been quantifying THC for a long time. The challenge is that the methods that are used for that are rather slow when you have to do literally hundreds or thousands of samples that are coming in. And so although the methods work very, very well, when you've got a huge volume of samples, it doesn't work that well at that stage. And so there's a dire need that crime labs have in order to address things like sample testing backlogs, the time-consuming nature of the experiments, the heavy resources that are used, both in terms of paying staff to run these samples or the consumables that are used. There's a dire need for there to be very rapid methods so that crime labs can do triage and reserve their resources for only those samples that really are marijuana so that then they can quantify those. So this is one of the big challenges, and this is one of the challenges that we're tackling.
Yeah. Well, on that very topic, could you tell us a bit more about the DART-HRMS approach that you previously mentioned? How did you develop it, and what are the most significant technical hurdles that you faced?
Well, DART-HRMS stands for Direct Analysis in Real Time, High Resolution Mass Spectrometry, and I'll be referring to that as DART-MS from here on out. It's a mass spectrometry method, and it's an ambient ionization method, which in contrast to the mass spectrometry methods that operate under high vacuum, it means that you can operate at atmospheric temperature and pressure, and you can do so out in open air, which makes the sample analysis very simple, but it also means that you can analyze samples in their native form. So one of the challenges that crime labs have currently with many of the approaches that they use, the conventional approaches, is that there's a lot of sample pretreatment that you have to have in order to render the samples into a form that makes them amenable to analysis by the conventional techniques. Whereas with DART-MS, you can simply analyze the sample in its native form. That's one. Two, you can get a spectrum within 3 to 5 seconds, which means that if you want to apply statistical analysis to data that you generate, or if you want to create a database of chemical signatures, for example, you need literally hundreds or thousands of those. to build a robust database. And so it's very, very helpful to be able to generate those signatures very quickly. So DART-MS allows you to do that. Another element of the technique is that in a really, really wonderful way, it allows you to get information on a range of small molecules or metabolites that are in the plant material. And because of that, you are able to see very polar compounds, you're able to detect very nonpolar compounds, and so it's a high information content approach, which is wonderful when you want to do statistical analysis, especially when you are trying to figure out which particular subset of molecules are important to being able to distinguish one type of sample or one class of sample from another, which is exactly what we want to do with hemp and marijuana. So these are some of the factors that make it really important for us.
And that follows on perfectly to my next question, which is how does the DART-HRMS and statistical analysis approach you developed revolutionize current practices in forensic laboratories regarding cannabis analysis?
It provides a mechanism by which many of the challenges that have slowed the analysis down in crime labs can be sped up dramatically. It provides cost savings, it provides time savings, because everything can be done a lot more quickly, and resources can be devoted to those samples that really are marijuana. And so I would say that this is one of the most important aspects of it. Yeah.
Brilliant, thank you. And could you discuss the role of advanced statistical processing in your research and its significance in improving the accuracy of differentiating hemp and marijuana?
You know, when we think about this issue, the statistical analysis, one of the ways in which that has manifested itself in forensic science is with DNA technology, right? So the chances of me and you having the same DNA profile I think are about 1 in 6.5 million or something ridiculous like that, which basically means that you're not going to get that wrong if you have the profile. It would be so wonderful if we could get those kinds of numbers for other types of evidence. Now, that's going to be very, very difficult, but at least it's a good idea if you can apply a certain level of certainty to the identification that you make. So when we have these chemical signatures that are attributable to hemp and marijuana, it's wonderful if we are able to say, we are 100% sure that this sample is hemp, or we're only 92.3% sure that this sample is marijuana. It's great to be able to do that. And the application of statistical analysis to the metabolome profiles allows us to be able to identify materials, but also associate the identification with a level of certainty, and to do so in a more objective way. And so this is very, very important when the stakes are so high, because people potentially can go to jail. And it's also important for exonerating people who are innocent. So I think this is one of the most important aspects.
Yeah, that's incredibly significant. Yeah. Thank you. So in one of your studies, you identified several values corresponding to known cannabinoids or major fragments from those molecules. How does this knowledge contribute to the broader understanding of cannabis? So
while it's true that hemp has generally lower levels of delta-9 THC, than marijuana, than the drug that's classified as marijuana, it's also important to understand the other ways in which the two plants differ, because the more you understand about that, the better able you are to have confirmatory testing to really distinguish between the two. And also it opens the door to finding cheaper ways to distinguish between the two. So in our case, for example, we've observed that there are differences in the terpenes that are present. And so it would be wonderful, for example, if we could start to correlate the differences in terpene profiles to the distinctions in the amounts of THC that are present in the two plants, because that can lead perhaps, for example, to cheaper ways, more cost-effective ways to test for the two and distinguish between them. And so those findings about other molecules that distinguish between them become very important in that context.
Yeah, brilliant, thank you. And based on your research findings, what are the next steps in addressing the challenges that crime labs face due to the legal distinction between hemp and marijuana?
Well, the first thing that has to happen is for people like us and others who are doing this kind of work, it's important that we publish the results so that the work is vetted. And then at that stage, if criminologists can start to use this to solve crimes, and this can be an accepted part of evidence that is entered into trials, then at that stage, courts will be much more comfortable. The judicial system will be a lot more comfortable using this technology. Forensic scientists tend to not want to change very much. And that's not really a criticism. There's a reason for it. The reason is that you want to fall back to what's tried and true, because ultimately you want to prosecute cases and you want to win those cases. And so you want to go with whatever's the gold standard in terms of what's been accepted by the courts. And so what needs to happen essentially is that if you've got a case, and in that case you really needed DART-MS data, or that was the best kind of data to use to prove the points or to prove the identity of materials, once that then gets accepted into the courts, then it becomes much more likely that more and more crime labs will embrace the technology so that it can be used, again, to exonerate the innocent and also to deal with those who've committed crimes. So I think that that's the next level, yeah.
Pittcon Perspectives, unforgettable Pittcon moments from the experts.
So, as we mark the 75th anniversary of Pittcon, could you share your first memory of attending this conference and how it impacted your view of the scientific community?
Yes, so I first attended Pittcon, I think it was in 2017. And it's a wonderful conference. It's the premier conference in analytical chemistry, if I'm not mistaken, on planet Earth. At least that's my opinion of it. And I go to a lot of conferences. One of the things that distinguish Pittcon, aside from the fact that you have, the presenters are well known, they're well established, they're doing cutting edge research, and so there's all the learning that comes from that. But one of the most amazing aspects for me is the presence of the vendors. And there are vendors at most conferences, but the vendors that come here bring their equipment with them, and it's running, it's on site. If you have samples, they will actually analyze them for you. And I have to say that for most of I've attended every year since 2017, and every time I come, I learn something new, some new cutting edge technology from a company. And usually we make purchases because it helps to further our research. So it's a fantastic conference from that perspective. So there's that element. Also, it's an opportunity for the individuals that I have the privilege of mentoring, whether they're postdocs or whether they're graduate students. It's an opportunity for them to come here and see and meet the people who wrote the textbooks that we're teaching from, talk to them, learn from practitioners about how their own research interfaces with the research that's going on out there, and how to make the necessary modifications so that it aligns with what practitioners want to use. For example, in the case of forensic scientists. So in an academic lab, we may come up with approaches, and the approaches may work wonderfully at the bench, but then the practitioner says, this is really cool, but it won't be well integrated into our forensic science workflows. But if you make this tweak or that tweak, this is how that could happen. And so that's really, really valuable information that allows us to translate our research into something that can be used by practitioners out in the world. So these are some of the wonderful things about this conference. Yes.
Mentorship is something that Pittcon is really passionate about cultivating and promoting. What has it meant to you to be a mentor in your work?
Oh my goodness. The closest that I may ever come to changing the world in this huge, dramatic way is likely going to be through one of these dear souls that I have the privilege of mentoring. And so I am heavily invested in their success. And I tell them, I'm your biggest fan. I want to mentor them in a way that allows them to fly solo so that they could get out into the world and do their own thing. And conference attendance is an immense, very important aspect of that mentoring experience. And so what I tend to do is it can be quite scary because you come here and if you're meeting people who wrote the textbooks from which you're learning, it's like, do I dare give a talk in front of this person? And so the mentoring process for me, aside from them being engaged in research, at school, at the academic institution, is to first introduce them to smaller local or regional conferences so that they can cut their teeth on that, and then help them to understand the extent to which their work is important and is of interest to everybody else in the field. And so it's when they come here that they get to experience that. And they're in a room and with people who actually showed up to hear what they had to say. And from the questions, I tell them, the questions you get, no one's trying to be mean. They're asking the questions not to test you, but because they're really interested in what you have to say and how it might impact their thinking on the work that they're doing. And so it's so important that you have that experience. So Pittcon for the students in my lab has been an immense aspect of the way in which I'm able to successfully mentor these students. And then for the past many years, several of my students, through exposure to Pittcon and exposure to the people here, have actually gotten jobs. So from start to finish, it's absolutely fantastic. And that's what we want, to earn a livelihood by our calling. And so it's a fantastic opportunity in that way. Yeah.
That's fantastic. Thank you. And finally, what are you most looking forward to Pittcon 2024 in San Diego?
Well, going out and seeing what the vendors have. I'm very, very interested in that. We're moving into some new areas. This is an analytical chemistry conference. Ultimately, one of the areas that we're moving into is cannabis intoxication, how you detect that. And there's so much to learn. There are a number of people here who are also in that space. And so there are opportunities to learn from them about how to accomplish that. There are some new findings that we have in my own lab that we're a little confused about. So we're thinking that other people may have worked on some of these issues, like some of the, you alluded earlier to some of the molecules or M/Z values, the masses that we're detecting by mass spectrometry that help us differentiate between hemp and marijuana. And it looks like there are other people who are in that space. So I'm really eager to see what they've discovered. And then also, I have a postdoc and a graduate student here with me from my lab, and I am eager to expose them to what's here. They're also giving talks, which is fantastic. It's a big deal to be able to be a student or a postdoc and give a talk here at Pittcon. And so it actually speaks to the importance of their work. So I'm excited about all of those dimensions and really, really happy to be here.